right, welcome everyone, and thank you for joining us today. My name is Angela. I'd like to welcome you to our webinar titled Improving Speed, Efficiency, and Resilience in the Development of Biopharmaceuticals with End-to-End Bioprocess Digital Twins. Before we get started, I just want to go over a few logistical details with you. So this is what the 23 webinar platform should look like on your end, with the screen on the left-hand side where you're currently seeing the presentation. And on the right-hand side here, you should see the questions panel at the top. This is where you can ask questions for the speaker, and then at the end of the webinar, we will have a Q&A session where we will respond to those. If you are joining us from a mobile phone, you should see a similar interface, but the questions box will be at the bottom and the screen will be available at the top for your viewing. And as I mentioned, please submit your questions for our presenter anytime during the webinar, so we'll have some time to go through them before the Q&A session at the end. Okay, and now I'd like to introduce our speaker for today. Dr. Edward Close is the head of the Bioprocess Modeling Competency Center at Siemens Digital Industries Software. He has an engineering doctorate in biochemical engineering from University College London in collaboration with Pfizer. Edward has over 10 years' experience at Siemens, leading the development and application of Siemens mechanistic and hybrid modeling solutions to deliver value across the pharmaceutical industry. Okay, so with that introduction, I'm going to go ahead and hand it over to Ed to get started on today's presentation.
Okay, well, thank you very much for the introduction, and thanks a lot everyone for joining. So welcome to today's webinar. So you've seen the title already. So let's get going. So let me start by asking a question. So why Bioprocess Digital Twins? So what we're hearing repeatedly is the biopharmaceutical industry is rapidly evolving. There's growing patient demand, there's new modalities, and there's a continual pressure to deliver. So there's an ongoing need to bring high quality biologics to the market faster, more efficiently, with flexible and sustainable processes, whilst still meeting regulatory expectations and business objectives. And of course, this is a very complex and difficult challenge. And what we see is organizations are increasingly looking to digital transformation to help them meet these challenges. So what we've seen is there's been a huge increase in demand for digitalization technologies in recent years. And in particular, the use of modeling and simulation stands out. So this is reflected in communications from academia, from presentations you see at industry conferences, and also now from regulators. So this is a nice quote from the FDA stating that in the last decade, modeling and simulation has become firmly established as a regulatory science priority. And this is a very nice publication from a few years ago, but if you go here, it kind of describes from their perspective successes and opportunities in modeling and simulation for the FDA. Industry agrees. This is a nice publication by industry leaders from 14 major biopharmaceutical manufacturers and suppliers. And it describes a remarkable agreement that they have on the problems to solve in industry and the significant opportunity to transform development and manufacturing through end-to-end integration and technology advancement. And this is really what we're going to be talking about in today's webinar. So at Siemens, we provide software, services, and online solutions that enable our customers to rapidly configure, calibrate, and deploy science-based, data-calibrated digital process twins. And I'm part of the bioprocess modeling team. I head up this team for a few years now. And before I go into all the details, I thought I'd first of all start with some basics. So you may ask, what is a bioprocess digital twin? So for us, bioprocess digital twins are comprehensive virtual representations of the bioprocesses that are utilized across development, engineering, and manufacturing to understand, to predict, and to optimize the physical counterpart's performance, to monitor it, and to facilitate decision making. And at Siemens, we have an end-to-end solution for bioprocess digital twins that is underpinned by science-based mechanistic and hybrid modeling approaches. So this is something that we've created in collaboration with big pharma partners for modeling the entire biomanufacturing process in a single platform that's used across the process lifecycle. So when we talk about end-to-end solution, first of all, we mean modeling the full bioprocess, so all of the different unit operations. And then secondly, we mean the ability to use those models integrated with each other to model the full bioprocess, and then to be able to take those models and use them across the process lifecycle. So why are people interested in this? So these bioprocess digital twins enable organizations to develop and produce medicines faster and cheaper by replacing physical experimentation with in silico modeling to design, to optimize, and to scale up biopharmaceutical manufacturing processes, and in the process reduce staff, material, and lab costs per project, which means that you have a more efficient organization and you can push more projects through the pipeline with the same resources. Then what we can do is we can deploy bioprocess digital twins into manufacturing for soft sensing, monitoring, and real-time optimization. And of course, this can then increase robustness, yield, and productivity. Oh, I think we have a little bit of noise on the background. Maybe that's you, Angela. Just before we jump into the first poll. So the term digital process twin is often used quite loosely. So I think it's useful to define a few categories that depend on the degree to which data flows from the physical object to the digital object. And so we have these three sort of categories: digital model, digital shadow, and digital twin. So with a digital model, the data flow is manual. With digital shadows, the data flows from the physical to the digital automatically, but the digital to the physical is still manual. And then we have the full digital twin where the data flow is completely automated. So what we tend to see in general is that in R&D and engineering, organizations are predominantly utilizing digital models and digital shadows. And then in manufacturing, it's more aligned with digital shadows and digital twins. However, there are certainly outliers. So for example, there are definitely cases where digital models are used by manufacturing to analyze deviations. And certainly I've had conversations where there's interest, at least, in utilizing digital twins for automated process development, where we have a back and forth between the experimental system and the digital twin, which is kind of guiding the actual experiments that are performed.
All right, so then we can go into our first poll. So we do have a few polls we're going to go show you guys throughout the webinar. So we'd appreciate if you guys would take the time to respond. So I just went ahead and started the poll. You guys should be seeing it now. And I'll leave it open for about 30 seconds or so for everybody to take the time to respond. All right, fantastic. So it looks like we got a lot of responses, so I'll go ahead and close that. And yeah, feel free to continue it.
Okay, so here at Siemens, we have customers that are utilizing digital process twins in bioprocessing, both offline and online. And so we have examples of digital models, digital shadows, and digital twins. So what we're going to do now is we're going to have a look at a number of examples. And the first thing we're going to look at is offline usage. And this tends to be where organizations kind of start their digital twin journey. Okay. So let's consider a bioprocess. So here we have a continuous monoclonal antibody manufacturing process. But what I'm going to describe in the next few slides is equally applicable to batch processes, and it also applies to the kind of range of modalities that we usually see across the industry, such as vaccines, other therapeutic proteins, gene therapies, mRNA, et cetera, So when we design these processes, unit operations are usually considered discretely, and this means they each receive their own set of control elements and acceptable ranges, which are then assembled together to provide an overall control strategy for the entire process. And I think historically, or at least over the last kind of decade, a lot of the applications where we see science-based mechanistic models, at least in biopharma, R&D, engineering, manufacturing, tends to focus on a single unit operation in the process chain at a time. And here, when we discuss this with our customers, we're always discussing sort of the advantages over a purely experimental approach. So being able to perform development tasks faster, cheaper, and with greater process understanding or fundamental understanding. So let's have a look at a few case studies here. So the first example is the design and optimization of a chromatography gradient solution. So this is a process that's producing a peptide and a oligonucleotide. And the step is one of the polishing steps that removes product-related impurities. And in particular, there's one which is particularly tricky. It's product impurity N-1. And this is because it elutes at a very similar time to the target. So we can also see here there's an N+1, a low charge, and high charge, which are also important. Now, optimizing this process experimentally required over 3 months and a significantly large experimental effort using a kind of traditional DOE approach. Now, in this project, a mechanistic chromatography model was calibrated using data from 3 gradient elution experiments. So these were linear gradients, and samples were taken every 1 column volume during the elution. And the concentration of each of the main species that we wanted to model was determined using HPLC. So this data was imported into the software and used to calibrate the adsorption isotherm part of the model. So in this particular case, we're using a rate model of chromatography. And the calibrated model was then used to explore the design space. And what we found was that the model was able to identify the optimal gradient in about a week. This was confirmed experimentally. Fascinatingly, the optimized gradient that the model produced was very similar to the experimental approach. But of course, it was able to identify that using significantly less material with only 3 experiments and in a much quicker time. So by introducing mechanistic modeling into development workflows, we're able to significantly reduce the number of experiments that we need to conduct, the time and the cost. Okay, so the second example is the optimization of the feeding strategy of a production bioreactor that produces monoclonal antibody. So this is a highly complex process, of course. There's lots of decision variables. In this particular case, there were 3 nutrient feeds that were fed daily and a temperature shift. So in this work, we start by configuring a mechanistic bioreactor model. And this includes a segregated cell population model where we're tracking multiple populations of cells. So we have viable cycling, viable resting, dead cells, and lyzed cells. And then we also have phenomena models to model the cell growth, the cell death, the cell lysis, the consumption of nutrients, the secretion of metabolites, and of course the production of the product of interest. So in this case, there were 22 experiments. So 7 experiments were used to calibrate the mechanistic model, and the remaining 15 were used for external model validation. And you're seeing here one of those, or part of one of those model validation runs. So when you're calibrating a bioreactor model, we not only need to know information about the cells, but we also need to know the data on things like the key nutrients and any toxic metabolites. And so in this case, we're using 1 sample per day. So for the majority of the experiments, we saw very good predictions. But in this case, we identified in the sort of analysis of the model validation that the model was struggling with high seeding densities and with the impact of temperature on cell culture kinetics. And in this case, the data which we collected for the model calibration was something that had been previously generated for sort of a DOE. And so this wasn't data that was designed specifically for model calibration. So it's important to be aware of this. So for this phenomenon, these phenomena, we explored a number of options. So the first thing that we explore is can we extend the mechanistic model, introduce some additional equations that would be able to describe that phenomenon. But in this case, we decided that actually this was part of the process that we didn't have enough fundamental understanding on, and so we moved to a hybrid modeling approach. So hybrid modeling is something which is reasonably new, and it's one of the big reasons why you now see bioprocess modeling in industry, whereas previously we didn't see it as often. And that's because we can use a hybrid approach where we embed something like an artificial neural network or even a PLS model sometimes into the mechanistic model to supplement the mechanistic model approach. So in this case, we have a parallel approach. So the inputs to the data-driven model and the kinetic model are the same. So for both of these, we're sending the concentration of the nutrients and the metabolites, the cell concentrations to both the data-driven model and the kinetic model. The kinetic model, which is fully mechanistic, predicts the rates of things like secretion, of growth, of death. And then the neural network, which is coupled very closely to that model and produces a correction factor. And that correction factor typically is something which is quite close to 1. And we can see on this graph here that this neural network producing a correction factor. So using this approach, we can get a much better prediction for the difficult to predict experiments. And so here we can see good fitting is maintained for the runs that were used previously to train the purely mechanistic model. And then we've introduced some additional runs to be able to calibrate the hybrid model. So this is something where we have now got a much better fit, but of course this is a fitting, so we expect this anyway. On the right-hand side, you see though the external model validation or the blind testing, and you can see here that we found that the prediction capability has improved greatly for a high seed density run that could not be predicted previously. Then the validated model was used to explore the optimal feed strategy, and in particular in this case, we also considered the usual variability that we would expect to see in this bioreactor. So rather than a point prediction where we look at one input and then we predict the outcome, instead we use the expected variability in the inputs, and we predict how that process will operate across that range of inputs. And you can see here that the model predicted that the biocapacitance feed strategy was something that would be more robust and have a better performance than the fixed feed. And this was also this was confirmed experimentally. Okay, the last example for this part of the presentation is a single pass tangential flow filtration. And here a mechanistic model was calibrated, and then it was used to predict the behavior of this process during flow rate changes expected during manufacturing. So on the top right-hand side, we can see that the model was able to match the experimental values very well. And then the two contour plots on the bottom here show another very useful application of modeling during development, which is to explore the design space. So these were design space studies that were used to provide supporting evidence of process robustness. And we're going to come back later to this particular case study. So you may ask, well, how did you do that? How is this all done? So the models that we're using here are all based upon known science and engineering principles to predict the CQAs from the CPPs that influence them. And I think that the real difference to point out is that whereas a data-driven model or a statistical model may have tens of equations, these mechanistic and hybrid models, which we're using, typically have tens of thousands of equations. So they tend to come with a huge amount of knowledge that kind of built into the structure of the equations. But of course, they're correspondingly more complex to build. Now that's where Siemens comes in. So what we've done is we've implemented a established library of integrated mechanistic and hybrid mathematical models of all the different unit operations that you would usually expect to see in bioprocessing. So that's the equipment and the different phenomena. And this has been based upon the very best published science available. And we established this through our opinions, but also the opinions of leading academics and of our kind of partners and customers. So for all of these models, the equations are derived from the natural laws of physics, chemistry, and biology. So they contain really deep mathematical knowledge of the process, which significantly reduces the data requirements. If you think about it, what you're doing here is you're leveraging many people's research into how processes actually work. So with these models, the parameters of the models tend to have a physical meaning. So opposed to a data-driven model where the parameters of the models often you can't directly attribute to something going on in the process. So a model parameter for chromatography might be the capacity of the resin. Then the other thing which is quite different is that these equations are typically differential equations, or things are based upon differential equations. And so what we're doing here is we're predicting and simulating the process dynamics. And this is quite different to statistical models, which are often simply an input-output correlation. Now, where there are gaps in our scientific knowledge, which often occurs, right, with bioprocesses, this means that sometimes we can't derive mechanistic equations or first principle equations for the entire unit operation or bioprocess. And this is where we've seen that big advancement recently with the creation of this hybrid modeling approach, where we embed data-driven elements such as artificial neural networks to fill in the gaps in our knowledge. And we do this in a way that preserves as much as possible the benefits of the mechanistic model. So we really try to use mechanistic equations as much as possible, and then only bring in the data-driven part to cover the gaps in our knowledge. So
Thank you for watching. To learn more about this topic, check out the links in the description. And if you enjoyed what you saw, like the video and subscribe to the channel.